Welcome back to our last lesson on microorganisms, where we'll wrap it up by looking at some scientific contributions and useful things that microorganisms do for us. So on page 45 of your textbook, two scientists are mentioned that have contributed to the field of microorganism studies, Alexander Fleming and Louis Pasteur. So Alexander Fleming first. He's the guy that is accredited with discovering the first antibiotic known as penicillin, and he did this really much by chance. Alexander is a pharmacologist, which means if you look at the keywords on page 45, that is a person who studies how drugs work on the human body. And he was actually investigating bacterial growth in a petri dish in his laboratory, which is this thing here. You can see the bacteria growing everywhere where there's some white parts. And he went on holiday. He went away for the weekend, locking up his lab. And when he got back after the weekend, he saw that on this petri dish, some of the bacteria have actually died, and in its place, a fungus has started to grow. So he deduced that the fungus that has taken up the residence of the petri dish now is probably producing some other chemical that is killing the bacteria in its wake. And that's what he isolated from it. Penicillin. Now, the reason he called it penicillin is because it's caused by a fungus that's called penicillium. So you can hear that the name for the drug is actually derived from the fungus that he discovered it from. So it's really interesting that fungi and bacteria actually produce chemicals like this, but it makes a lot of sense. Remember that on the surface of your skin at this very moment, you are covered with microorganisms that you're not even aware of. There are viruses and bacteria and fungi, all of which are competing for your skin. They're competing for a place to stay and grow. So it makes sense that microorganisms make chemicals against each other that they use to fight each other, the same way we make weapons to kill each other. So all that we as humans have actually done is we have isolated these chemicals and we've called them antibiotics because they help us to kill bacteria. Louis Pasteur is most well known for his discovery of pasteurization, or his invention rather, of the process of pasteurization that helps us to elongate the shelf life of things like milk, for example. But he's also known for his discoveries in the production of vaccines against rabies and anthrax, and he's also well known for being the first one to actually say that germs cause disease. Now, you have to know what exactly pasteurization entails. So on page 45, it says that pasteurization is a process where, for example, the long-life milk that we buy in stores is very quickly heated to about 72 degrees for 15 seconds and then quickly cooled down again. And the process of rapidly heating and cooling this milk will help to eliminate microorganisms and therefore elongate the shelf life of that milk. So that's why we refer to milk like this as long life milk. When you ultimately open that milk in your kitchen and start using it, bacteria can gain access to the milk inside the container. And so ultimately long life milk will eventually go sour as well. Now, microorganisms are also useful, all right? And on page 46 of your textbook, it only mentions one use of microorganisms, namely yeast in the process of bread making. But we want you to make a note of two other reasons that microorganisms are important for us. The first of which is that they are decomposers in ecosystems. Now, if you've never thought of this before, take a moment to think about what life would be like if we never had decomposers. You would still be sitting amongst every piece of fruit, every piece of bread, every piece of any food or any clothing or any piece of paper that you've ever tossed in a bin. You would still have that around. Dumpsters would be much larger and there would be much more waste that is not decomposing. So decomposers are mainly microorganisms. Bacteria and fungi help us to recycle nutrients by breaking that dead plant and animal material down and putting those nutrients back into the soil. So that's the first and most important factor that they actually play in our lives. The second reason why microorganisms are useful is for our own digestive processes. Inside our large intestine, there are naturally occurring bacteria that have a symbiotic relationship with us. We give it a nice toasty environment to stay in, where it gets a lot of feces to feed on, and in exchange for that, it provides us with vitamin K. Vitamin K is essential for the clotting of blood, so we actually need this bacteria in our intestines to stay healthy. The third thing that microorganisms are useful for is actually mentioned on page 46, which is yeast that's used, for example, in the baking of bread, which means fermentation. Fermentation is the third important thing that microorganisms provide us with. Fermentation is a process where sugars are broken down to produce alcohol and carbon dioxide. All of us that have baked bread, or maybe you made some ginger beer during the lockdown, you've all experienced working with yeast in the kitchen. You might have thought that the yeast you buy in that little packet looks like a kind of a powder or some granules of some kind, but that's actually a living organism. It's a type of fungus that's just sleeping because it's not activated in that packet. When you put the yeast into the bowl and you add your lukewarm water and the sugar or the flour that you're using to make the dough, you're actually activating the yeast. You're waking it up and telling it, oh, here is something for you to digest. So the yeast will then start changing the sugar and the starch within the flour into alcohol and carbon dioxide. That carbon dioxide is what causes the bread dough to rise. It puts air into the dough and creates the spongy texture of the bread. The alcohol that it produces, you can actually taste and smell if you've ever done that with raw bread dough. But as soon as you put the bread into the oven and you start baking it, that yeast will die and the alcohol will evaporate. So the bread that you take out of the oven will have a nice spongy texture because of the carbon dioxide, but the alcohol and the yeast will no longer be present in the bread. However, if you're using yeast to make wine and beer, that fermentation process keeps the alcohol inside the compound that you're producing, because that's the whole point, right? We want to produce an alcoholic beverage. You can also see the bubbles here inside the beer. That's obviously the carbon dioxide that the yeast has produced inside the beer. So fermentation is the third important thing that microorganisms do for us. So that's it for this chapter, and you can now complete exercise 3 at the end of the chapter on page 48.